everyone. Myself Manju Sharma from Suresh Kanvyar University, Jagatpura, Jaipur. Department is School of Agriculture. Today we are going to start about diseases of sapota. So here we are discussing about major diseases of sapota. Basically, there are three diseases: flat limb, leaf spot, and sooty mold disease. So flat limb, which goes by Botrytis diplodia and Theobromia; leaf spot, which goes by Phaeophyllosspora indica; and third one is the sooty mole disease, which goes by Capnodium versicolor. Flat limb, the causal organism is Botrytis Theobromine, the first reported from Maharashtra and Gujarat, now prevalent in all sapota growing areas of the country. The disease is a problem in orchards about 10 years age. Symptoms: branches of affected trees become flat and twisted at the terminus, leaves become thin, small, and yellow and are produced in clusters on the affected branches. Very few flowers are produced on affected branches, which will remain infertile. Fruit set is rare, and when set, fruits are undersized, hard, and fail to ripen. Then foliage and fruits fall prematurely. Survival and spread, like primary conidia or infected plants, secondary wind-borne conidia, then their management. Pruning of affected branches, which followed by copper oxychloride or Captain Orginab 0.3% spray. Next disease is the leaf spot disease, which goes by Phaeophyllosspora indica or Pestalotiopsis versicolor. Disease symptoms are like earlier circular spots which pinkish, then gradually to brownish in color, and the center of the spot sometimes whitish gray color, and number of spots are more on leaves. Then Pestalotiopsis versicolor. So the spores are circular and brownish and bigger. Later stages can see the black dots on center of the spore. These black dots are the asexual fruiting body of the fungus, that is acerulus. In advanced stages leads to defoliation. Numerous small, circular, pinkish to reddish brown, conspicuous spots with whitish center on mature leaves. Then the spots are coleus and leaves drop prematurely. Survival and spread. The pathogen is seed-borne fungus. An inoculum present in the seeds are source of primary infection. Fungus also survive on glumes, fruits, and plant debris. Favorable conditions. Rainy season favors the development of disease. Their management: carbendazim 0.1% and companion, that is combi product, includes carbendazim and mancozeb 12% and 72% to avoid resistant development in pathogen. Then the next disease is the sooty mole disease, which caused by the Capnodium versicolor. The disease symptoms are: it is a fungal disease developed on honeydew-like excretion secreted by aphids and scale insects. Disease severity increases in increased population of leafhoppers, aphids and other insects, black superficial growth on entire surface of leaves, roots and twigs. Fungus is not a parasite. It grows on the excreta and honey secretions of insects. The fungus slowly covers the entire leaf area, severely affecting the process of photosynthesis. So, under dry spell, such affected leaf curl and shrivel. During flowering time, the appearance of the disease results in reduced fruit set. Then, the sooty mass is a superficial growth of the fungus and it multiplies on insect secretions. This result in reduced translocation of food to the fruits, which leads to reduction in their size. Then, the impact of this disease on host is photosynthesis activity and yield decreases. Basically, these are the symptoms of the disease. Like the color of the leaves will be changed, survival and spread. The severity of infection depends on the honeydew secretion by the scale insects that provide the necessary medium for the fungal growth. Then transmission occurs by airborne ascospores. Favorable conditions like high humidity and moist situation favors the development of disease. Their management, like we have to spray off wettable sulfur 0.2% along with insecticide dimethoate, and it should be 1.5 gram per liter, and spray of 1% starch solution makes flags on the fungus and due to small windfalls from the plant. So this is all about the major diseases of sapota. Now the next is the diseases of coconut. Coconut, the scientific name of the coconut is Cocos nucifera. The family is Adiaceae. The coconut palm is affected by a number of diseases, some of which are lethal, while others gradually reduce the vigor of the palm, causing severe loss in yield. So, a brief account of the important coconut diseases is given. First is the bud rot. The causal organism is Phytophthora palmivora. Economic importance are, like this is one of the serious disease of the coconut and is fatal since the growing part of the plant is damaged. Bud rot incidence is high in palm growing in marshy, waterlogging areas and in environments that promote high humidity above 90%. So the incidence is mostly found to be severe during monsoon when the relative humidity is very high and temperature is about 24 degrees centigrade. The symptoms are: the earlier symptom is the yellowing of one or two younger leaves. Black spot appear on spindle leaves. In the later stages, the spindle whitens and drop down, so the tender leaf base and soft tissues of the crown rot into a slimy mass of decayed material emitting foul smell. Then ultimately the entire crown falls down and the palm will be dies. So basically these are the symptoms. One plant which is a healthy plant and another one is infected plant. Primary source of inoculum or inoculation like dormant mycelia or oospores. Secondary source of inoculation, these are the zoospores and sporangia, and mode of actions are throat stomata. Epidemiology, like cool weather loving fungi, temperature should be 18 to 20 degrees centigrade, relative humidity 100%, closed plant population, time of southwest monsoon, and susceptible host. These all the factors which are necessary for the pathogen or to cause the disease in the plant. So that is called the epidemiology. Control: in the bud rot affected plantation, regular spraying with 1% Bordeaux mixture just before and after the monsoon to adjacent healthy palms, orchard sanitation by removal of dry material from the crown is also essential. So if the disease is detected in early stage itself, then application of Bordeaux paste on the crown after throughout cleaning and removal of infected material can check the disease. The treated portion should be given a protective covering with polythene sheet to prevent washing off of the paste. So it is best to remove infected palms and treat the surrounding palms. Trunk and root rot or Ganoderma wilt or Thanjavular wilt. The causal organism is Ganoderma lucidum. The economic importance like this malady was first noticed in Thanjavur district at Tamil Nadu after the cyclones of 1952 and 1955 and hence called Thanjavur wilt. Now it is widespread in other districts like Kanyakumari, Tiruchirappalli, South Arcot and North Arcot, and also the border areas of Kerala. The symptoms are: all stages of the palm growth are attacked by the pathogen. The fungus infects the roots and spread upward, killing the entire root system. Then the trunk tissues internally root and reddish brown gummy fluid exudes out from natural growth cracks. As a result, reddish brown lesions are formed on trunk starting from base. Then the infection progresses and lesions extend linearly and radically on trunk or infected palms. So, because of rotting of internal tissues, including the cortex and the vascular system, water and nutrient transport will be impeded. So, the older leaves droop and wither and remain suspended around the trunk. Infected tree dies in a few months of 2 years. Then, on palm with advanced infection, and on dead palms the fungus produce fruiting bodies, that is the basidiocarps, along the sides of the basal trunk, particularly during periods of heavy rains in the monsoon. So these are the disease symptoms. Basically, there are the many wounds by which it will be weak and the plant will be die. Survival and spread. Primary survival like basidiospores or saprophytic mycelium in soil and infected debris or several host tree species like mango, jackfruit, citrus, Eriacanet, poplar and several forest trees. Secondary, like mycelium and basidiospores, through irrigation water or root contact. Favorable conditions like dry soil and weather conditions, then the later external soil, particularly by the side of river beds, then newly cleared forest lands planted with either coconut or other susceptible tree crops. Their management. Cultural practices should be followed judiciously. Repeated plugging in infected areas may be minimized. The closer planting avoided, adequate irrigations need to be given, providing mulches or green manuring at the basin of the palm. Diseased palm should be removed and burnt. Bleeding patches should be chiseled completely, followed by hot coal tar application, then application of 5 kg neem, 5 kg neem cake along with sufficient organic matter and 500 gram phosphate plus 1200 gram potassic fertilizer may be applied per palm per year so as to keep proper nutrient in the soil. Drenching with 1% Bodex mixture and stem injection or root feeding of 2 gram Oreofungin solution and 1 gram copper sulfate in 100 ml thrice a year for only 1 year may reduce the intensity of this malady. Next is the stem bleeding. Next is stem bleeding, which goes by the ceratocystis paradoxa. The causal organism is ceratocystis paradoxa. Symptoms: trunks of tender palms are more attacked. Water shock patch on trunks appear, initially is inside. Reddish brown ooze from the cracks in the patch is a characteristic symptoms. So the fluid rise up to form black encrustation with brownish orange margins. So the patch progresses linearly upward direction when the severely decayed patches are pierced. A yellowish fermented fluid gushes out. So large cavities are formed in diseased trunk. Then the infected trunk are usually infested with Diocalendron beetle. Growth of affected palm is arrested and yield will be reduced. So these are the symptoms. Basically, there will be a liquid will be secreted from the stem of the coconut plant. That's why the name is stem bleeding. So there is exudation of reddish brown liquid throat cracks on a coconut trunk. Survival and spread: primary survival, perithecia, chlamydospores in cracks and cavities of infected stem and petioles of fallen leaves. Secondary, conidia dispersed by irrigation water, insect wind. Favorable conditions: heavy rainfall which followed by drought, high water table and highly alkaline soils. Then cracks and insects, wounds caused during harvest and leaf removal, and poor fertilization and poor maintenance of orchids. Management: avoid any mechanical injury to the stem, provide proper drainage during rainy season, chisel out affected tissues, hot coal tar or Bordeaux mixture in alder trees. After draining the fluid, after draining the fluid, cavities should be filled with a mixture of charcoal, sand, and cement to strengthen the trunk and to prevent the secondary damage. Coat the lower part of trunk with coal tar or Bordeaux paste to prevent the disease. Coconut stem boring: insects should be controlled by applying carbaryl 50% vegetable powder on the trunk at 3 gram per liter. Then the next disease is the Grey leaf blight or grey leaf spot. The causal organism is Pestalotia palmarum. The symptoms are leaf spot symptoms develop only on the mature leaves in the form of small yellowish brown spot on the leaflets which gradually become oval in shape with a grayish band in the advanced stage. The affected portion of the leaflets show a burnt or blighted appearance. So basically these are the symptoms of the disease. Survival and spread. Primary survivor conidia in infected plant debris. Secondary conidia dispersed by rain splash and wind. Favorable conditions like neglected garden with palms suffering from potassium deficiency, heavy weed infestation in the basin of palms. Their management: remove all the affected leaves and burn, correct potassium deficiency by applying recommended dose of potassium 3 kg per palm. Then keep the basins of plant weed free and spray foliage with Bordex mixture 1% or copper oxychloride or Captain 0.25%. So this is all about the diseases of coconut. Now, diseases of beans. The first disease is the anthracnose disease. The causal organism is Coletotrichum lindemutanium. The symptoms are all the above ground parts are affected at any stage of crop growth. However, the characteristic symptoms appears on pods. On cotyledons, spores are sunken, dark brown or black with pink spore mass. The seedling infection results in collapse of seedling. Spots on leaves appear on lower side and are black. Later these may also appear on upper surface. When the infection is severe, the affected plants whiter, black, sunken circular spot of varying size appear on pods with bright red, yellow or orange margins. Seedling infection results in collapse of seedling. Spores on leaves appear on lower side and are black. Later these may also appear on upper surface. So when the infection is severe, the affected plants whiter. Spores on leaves appear on lower side and are black. Later these may also appear on upper surface. So when the infection is severe, the affected plants whiter, black. Second, circular spots of varying sizes appear on pods with bright red, yellow, or orange margins. The center of these spores later turns gray or pink due to sporulation of the pathogen, and the border of these spores appear raised. These are the symptoms, basically spores on the beans. Survival and spread. Primary survival, mycelium or conidia on seed and collateral host, cowpea, mung bean, and black gram. Secondary, wind blown and rain splashed conidia. Management, use healthy seed for planting. Periodically harvest all mature pods are shot and destroy the infected ones. Then seed treatment with carbendazim 2 gram per kg of seed. Protect the crop by spraying banomyl or carbendazim 0.1% or Ginep or Mancozeb 0.25% at 10 to 15 days interval. Then the rust disease on beans, that is Euromyces phaseolitipica, on cowpea and others Euromyces fa- facilio vagni, and on dry bean Euromyces appendiculatus. Autoecious macrocyclic rust, which produce all the stages on bean plant. So this is the rust disease. Symptoms. The symptoms mostly appear on leaves, throat found on petiole and stem of some post. The rust pustules appear on either side of leaves but more common on lower surface. The uridosauri are minute, roundish, slightly raised and reddish brown in color. These contain rust spore or appear in the groups. Later in the season, with the formation of teliospores, the sorry turn dark brown or black. Then the diseased leaf may wither or fall off under severe infection. So basically, these are the symptoms: upper side of dry bean leaf with rust and the yellow holes. Like the first diagram, which is showing the dry bean leaf with the rust disease and yellow holes. Survival and spread. Primary survival, like survive throughout teleospores in cooler regions and on collateral hosts. Then the secondary, wind-borne uridospores. After that, their management, early showing to avoid severe infection of plant in the field. Their management, early showing to avoid severe infection of plant in the field and protect the crop with mancozeb or ginab 0.25% or the vegetable sulfur is 0.3%. The next disease is yellow mosaic, bean yellow mosaic virus, mung bean yellow mosaic virus, or fasciolus virus 2. virus, and there is the single-stranded DNA. The symptoms are bright yellow patches appear on leaves. Yellow areas alternate with dark green areas of the leaf blade. Younger leaves show more severe mottling and chlorosis. So the younger leaves show more severe mottling and chlorosis. Basically, bright yellow patches appear on leaves. Yellow areas alternate with dark green areas of the leaf blade. So the leaves completely turn yellow and gradually become necrotic. The plants are stunned and flower and pod set is reduced. Pod formation is reduced, and if produced, then they are deformed, having shriveled and undersized seeds and may exhibit mottling when green. So basically the one is the healthy plant and the another one is the diseased plant. So the color of the leaves which becomes turned white from green color. Survival and spread. Primary survival, like virions in collateral host. Virions in collateral host. French bean, soybean, red gram, Gentium estrumerum, Eclipta alba, croton species, black gram, green gram, etc. The secondary survival and spread. Virions transmitted by whitefly, Bemisia typica. Management: remove collateral host and destroy from the vicinity of the field. Use resistant varieties. Manage vector by erecting yellow sticky trap and spraying systemic insecticide, that is methyl dimetone or dimethoate, 2 ml per liter. Then common bean mosaic virus or green mosaic bean common mosaic virus. Common bean mosaic virus or green mosaic or bean common mosaic virus. So virus with long flaxeous rod having single stranded RNA. So the symptoms are affected only beans, that is Phleolus vulgaris and other. Leaves show mosaic pattern, like light green areas alternate with dark green areas. The diseased leaves become rough and show blisters on the leaf lamina. Leaf size, petiole length, and plant height reduced, and leaves curl downward. So the diseased leaves become rough and show blisters on the leaf lamina. Leaf size, petiole length, and plant height reduced. Leaves curl downward. Then the diseased plant produced for fever pods which are smaller in size. Then the seed becomes smaller, malformed and uprooted. One is the healthy plant symptom, and another one is the diseased plant symptoms. Survival and spread. Primary, like virions in seed and collateral host. Secondary, virions transmitted by aphids, Apis cruciferum, Marcosiphium, Apis gossypi, Apis medicagenis, Myzus persici, and Brevicoreni brassici. Then their management, use of disease-free seed collected from healthy plants. Then the vector management by raising barrier crops and spraying systemic fungicide, methyl dimethoate or dimethoate 2 ml per liter or acephate 1.5 gram per ml or imidacloprid 0.2 ml per liter, either alone or mixed with neem oil. So this is all about the diseases of beans, their survival and spread, symptoms and management. Thank you.